So that's kind of the big gross anatomy. Now let's look at really the nuts and bolts, the functional anatomy of the kidney. Why is this called the functional anatomy of the kidney? Because this is actually what is doing the work. Nephrons. It is the structural, it is the functional unit of the kidney. If you destroy a nephron, you destroy the functionality of the kidney. So that's why it's generally referred to as the functional unit. When we talk about the nephron, it's microscopic, so it's not part of the gross anatomy, which is what we just kind of discussed. But when we talk about the nephron, it's this glomerulus structure, this tubular network that's associated with it, and then the collecting duct. And we'll get into all of these little parts and pieces here, okay? Which kind of big picture, it's called renal corpuscle, which is this thing right here, and all of the tubes that are associated with that corpuscle. All of the corpuscles, and what that is just a knot of capillaries with some tissue around it. All of those things, all those corpuscles, they are all within the cortex. The tubular structures, as you can kind of see, and here's all the names of them. We'll talk about them in a second. Proximal convoluted tubule, distal convoluted tubule, the nephron loop. All of those, well, some of those are within the cortex and certain parts of them are within the medulla. So I guess you could say the vast majority of them are in the cortex, but that will become evident here in a little bit. So here is the renal corpuscle right there. Okay, there it is right there. Okay. The renal corpuscle consists of several different structures. Collectively, though, it's referred to as a glomerulus. The glomerulus, and then the glomerular capsule. So those are the two major parts. The glomerulus is one, and this is just, oh, good Lord, one. And this is just capillaries. That's all it is. And then the glomerular capsule is the second part. And it surrounds the nephron loop. If my hand here is the nephron loop, and my other hand here is reaching around it, there we go, okay? There you go, now you can see it, okay? So this is the nephron loop, and the capsule is what wraps around it. It has some certain pieces of anatomy that make it unique, that really help with the first step in urine formation, which happens here within the renal corpuscle itself. Blood enters into the renal corpuscle, and you can see it right here. Comes in from one of the many arteries, right? We don't need to worry about the name of that artery. This one you do, though. This artery here brings in blood to the glomerulus. That is the afferent arteriole. You can see it's really big compared to the efferent arteriole, which is right here. Efferent takes it away, just like sensory or afferent took it into the brain. Efferent motor took it away from the brain. So afferent and efferent are kind of the same thing here, going to and taking away. So afferent brings blood to the nephron. Efferent takes blood away from the nephron. And I can't remember if I have it listed, but there are two distinct things here that are happening. One, the afferent arteriole is a little bit bigger than the efferent arteriole, and that is significant, all right? Having that slightly smaller exit creates a pressure, right? Because if there's a lot of fluid coming in, but only a little bit can get out, that creates pressure within this tube, which is going to be fantastic for what it's designed to do, and that's to help initially form what we call filtrate. It's going to end up being urine, but it helps to form filtrate. So that one little piece of anatomy is significant. The glomerular capsule itself, and it doesn't really, I guess it does. I mean, the glomerular capsule, here's a picture here, and we'll have some pictures a little bit later, but the glomerular capsule has two parts to it. We have a parietal layer and a visceral layer. Parietal layer Any visceral layer? Haven't we seen parietal and visceral layers for everything from here so far? Good gosh, yes. So here's the parietal layer. I don't even know if they kind of show it off. Yeah, they have it labeled. Here's the parietal layer. Okay. And then it folds back and forms a visceral layer. And that visceral layer looks really weird. Okay. Those things are called podocytes. They are the cells that wrap over the capillary knot. That is the visceral layer of the glomerular capsule. We'll talk about that in a little bit. One simple squamous epithelium, some specialized simple squamous epithelium called podocytes. Don't worry, we'll get to it in a second. And then obviously we have a little space that you can kind of see here in between the two visceral and parietal layers. And that helps to receive filtrate. Okay, and that's what it does. Glomerulus, capillary knot. Glomerular capsule, and old school name is Bowman's, but they don't like us using that anymore. So glomerular capsule. The glomerular capsule has a parietal layer that lines the space and a visceral layer that lines the outer surface of the capillary bed, that capillary knot. There's a vascular pole and a tubular pole, as you can kind of see on here as well. Doesn't matter. Don't worry about it. As we leave the glomerulus, as we leave the renal corpuscle, I guess I should say, collectively, both structures, we enter into a tubular network. You have a tubular network that is first in line. That's the proximal convoluted tubule. It's the first region of the tube right here. Okay. Proximal convoluted tubule, it has some wiggle to it. Convolution. It's simple cuboidal epithelium, as you can kind of see from the cells there. So it's going to be involved in some secretion, some reabsorption. You can see from the picture there, it appears to have a brush border, and it does. It has a very extensive brush border, which is what allows us more surface area. Microvilli. Increases surface area for reabsorption. Okay. Increases surface area for reabsorption. Because I'll just give you a little spoiler. In the renal corpuscle, we basically push out everything. Fluid, nutrients, good, bad, evil, all of it gets shoved out. And then the body is monitoring the levels within the blood, and it's like, hey, I would like some of those back, please. So here in the proximal convoluted tubule, the nephron will reabsorb them back into the body, and we'll see how it does that here in a second as we lay over the blood vessels associated with these areas. We have the nephron loop. We have a descending nephron loop. We have an ascending nephron loop, and you can see there's a thick and a thin portion associated with it. Okay. There's a thick and a thin region associated with it. Sorry about that, guys. There's that. There is some significance into that, but we're not going to get into all of the specifics. That's at a much kind of higher level. But you can kind of see there, guys, with the nephron loop, you have one section where it is thicker. It's more simple cuboidal in nature. And then we have a section that's more thinner, and that's going to be more simple squamous in nature. And there is some significance into that, and we're not going to get into it. Certain things are going to be reabsorbed a little bit more easily in the thinner part than obviously in the little bit thicker part. The nephron loop does dive down either a little bit into the medulla, which is right here, okay, or it can do a deep dive deep into the medulla itself. And generally, that's just a nephron loop that does it. it'll have some significance in a little bit. And the last portion of the nephron is the distal convoluted tubule. The distal convoluted tubule is lined by simple cuboidal, just like the proximal is. But here, we don't have microvilli. So no microvilli. No micro, M-I-C-R-O-V-I-L-L-I. No microvilli here. And that's significant. There's not a lot of reabsorption that occurs here. There's usually a lot more secretion that occurs here. Can reabsorption occur here? Yes. Not as much though. Okay. And the distal convoluted tubule has some unique characteristics associated with it. So especially when it comes to reabsorbing certain things, and we'll hint at some of that later. The last portion is the collecting duct, but it is technically not part of the nephron, so we'll talk about that later. When we have nephrons, I keep making reference to going in a little bit into the medulla, going a whole lot into the medulla, and here's what that looks like. Nephrons, we have two different types. We have nephrons that are primarily completely within the cortex, called cortical nephrons. And we have nephrons that are in the cortex, but they go deep into the medulla, and those are called juxtamedullary. Juxta means in close proximity or in close position to. So these are nephrons that are in close proximity to the medulla, hence the name. Their names are based on, as you can kind of figure, where their renal corpuscle is. One's more higher within the cortex, and one's close to this juxtamedullary junction, whatever. I base it more on the nephron loop. If the nephron loop is completely within the medulla, it's a juxtamedullary. If the nephron loop is just a tiny bit in, then it's a cortical nephron. When we talk about nephrons, generally we talk about juxtamedullary. They are, as you can see, the bulk majority. I won't ask you that percentage. It's just there to impress upon you that these are the bulk majority. Okay. Why do they have a very long nephron loop? Because that's what's going to help us concentrate urine. What does that mean? Well, how much water do we hold on to, or how much water do we get rid of? That's what the juxtamedullary nephron does. So it helps to form a concentration gradient, a salt concentration gradient, NaCl. And this is going to allow us to affect the concentration of urine, i.e., how much water do I produce and get rid of? If I'm obviously not getting enough water, I'm going to reabsorb more of the water. If I'm drinking way too much water, then I'm going to produce a copious amount of urine, watery urine. And again, figuring out those levels is done by the juxtamedullary nephron. The last little bit, guys, after the distal convoluted tubule is the collecting duct. Nephrons drain into collecting tubules. This is the distal. I guess let's do it in order. Proximal convoluted tubule, the nephron loop, and the distal convoluted tubule. Okay, so that's their term for all of those tubes that we just talked about. All of these collecting tubules dump into a larger collecting duct. From the collecting duct to the renal papillae, to the minor calyx, to the major calyx, and out renal pelvis, ureter, bladder, urethra, toilet. There you go. Lining the distal convoluted tubule and lining the collecting tubules and lining the collecting duct, we do have some specialized cells. These are going to be cells that are very responsive to hormones, I guess I should say. They are very sensitive and they can be manipulated, especially the distal convoluted tubule and especially the collecting duct. Those two, more than any other duct or tube associated with the nephron, is manipulated a lot, those two. When we look at those two, we're going to see some specialized epithelial cells there, just like we saw specialized epithelial cells in the lungs. Pneumocyte type 1 and type 2 cells, or alveolar type 1 cells and alveolar type 2 cells. These are called principal cells and intercalated cells. These are both specialized epithelial cells, okay? They're just found within the distal convoluted tubule and the collecting duct. Principal cells are very responsive to aldosterone and antidiuretic hormone, affecting water reabsorption or water secretion, however you want to look at it. The intercalated cells, and there are two types, I'm not asking. These help to regulate the pH of your urine and the pH of blood, okay? Which is going to affect hydrogen ion concentration, which can affect CO2 level concentrations within the urine, within the blood, okay? So pretty similar in how the respiratory system regulated how fast our rate and depth of breathing occurred by CO2 levels, by hydrogen ion levels. Same thing here within the nephron. We have a few other cells as well that help us to basically form urine. Filtrate is the first thing formed before it's officially called urine. This is found, I'm going to go over here. You can see it also helps to regulate systemic blood pressure because it helps to regulate blood volume. But if I go here and look at a blowed up version of the renal corpuscle, here is the afferent arteriole coming in, big and thick, efferent arteriole leading away, a little bit narrower. So it helps to create that pressure that I talked about. Here you can see a nice parietal layer, and as it folds back, now you can see that podocyte a hell of a lot better. That's the visceral layer. But you can see we have some cells here called the juxtaglomerular apparatus, okay? And the juxtalar glomerular apparatus contains what are called granular cells and macula densa cells, okay? And here they are. I mean, that's where they are. I'm not going to ask you to identify them, okay? Macula densa cells are all concentrated right here. Why are they concentrated right there? Because they're affecting blood flow into the glomerulus. I don't care if it already exits. I care when it enters, because when it enters, then I can affect it. Water concentration, concentration of ions, all that fun stuff. And then granular cells are all right along that afferent arteriole right there. So collectively, we just refer to them as the juxtaglomerular apparatus. Juxta in close proximity to the glomerulus. As you can kind of see, I guess I'll underline this aspect, guys. These are modified smooth muscle, and this is modified epithelial cells, okay? This is in the afferent arteriole, and this is in the distal convoluted tubule. If I want to regulate the amount of blood flow into the glomerulus, then obviously I want to contract muscles, right? Well, specifically smooth muscle. That's the only one we have. So these granular cells are going to help regulate how much urine we form. It's going to affect what's called the glomerular filtration rate. So it's going to affect blood volume. It's going to affect blood pressure. They create the renin-angiotensin system that we talked about, a potent vasoconstrictor. So it's going to be involved with helping to regulate blood pressure. The macula densa cells, they are going to be found, they're modified epithelial, found in the distal convoluted tubule. And these guys detect changes in salt concentration. They are going to help with water levels. If I have too much salt, too little salt, it's the distal convoluted tubule and the collecting duct that will vary the water amount maintained within the urine. And this is what tells those granular cells to secrete the renin. Okay. It's called paracrine secretion. It's secretion to a nearby cell. That's all that means. Okay. So granular cells, what is it? It's modified smooth muscle. Where is it found? The afferent arteriole. What does it do? It's involved with regulating glomerular filtration. It helps to regulate blood pressure. Macula densa cell, what is it? Modified epithelial cell. Where is it found? Distal convoluted tubule. What does it regulate? Sodium chloride concentration. What does that help with? Helps with forming a more or less concentrated urine, depending upon my NaCl levels. It yells at the granular cells to secrete the renin, to vasoconstrict, to help restore blood pressure within the system. Because you're forming less urine, because you're filtering less water out of the blood. There's some other cells there. You can see there's some mesangial cells. And they don't truly fully understand how they function exactly. So we're not going to talk about those. Blood supply to the kidney, it is extensive, right? It is extensive.